Hey there, amazing people. I'm thrilled to have you here today. Welcome aboard as we set sail on a journey through Dale Sanders. Dale Sanders, FRS, born 13 May 1953, is a plant biologist and former director of the John Innes Centre. The centre is an institute for research in plant sciences and microbiology in Norwich, England. Let's now zoom in on education and uncover the hidden gems that lie within. Sanders was educated at the Hemel Hempstead School. He gained a Bachelor of Arts degree from the University of York, reading biology from 1971 to 1974, graduating with first-class honours. Sanders did his FT alongside Professor Enid A.C. Macroby FRS at Darwin College, Cambridge in 1978, in the Department of Plant Sciences. In 1993, Sanders earned his SCT from the University of Cambridge. As we venture forward, let's take a closer look at research and its impact on our understanding. Sanders' research explores the transport of ions across plant cell membranes and the roles of ions in signaling and nutrient status. Sanders' first significant finding during his FT was to provide unequivocal evidence that inorganic anion uptake in plants is powered by a proton gradient, and showed how transport is regulated through intracellular ion concentrations. In subsequent research as a postdoc at Yale University School of Medicine, he pioneered the first methods to measure and interpret the interplay between control of intracellular F and activity of the plasma membrane proton pump, showing how the regulation of the proton pump is controlled by and in turn controls intracellular F. This work on a fungus served as a paradigm for understanding the interplay of membrane transport and cellular homeostasis in fungal and plant cells. On taking an academic position at the University of York, Sanders developed novel electrophysiological approaches to plant cellular signaling and membrane transport. The Sanders lab demonstrated a key link between changes in cytosolic free calcium and photosynthetic activity, and through many technical developments showed how membrane transport at the plant vacuole is energized and regulated in response to physiological demand. Sanders also developed a unified mathematical theory that explained complex kinetics of solute uptake in plants, along with having created the first methodology to measure transient changes in intracellular calcium levels in higher plants, and discovered that light arc changes in photosynthetic activity were highly dependent on cytosolic changes in calcium. In the days before extensive molecular biology, Sanders discovered that the vacuole proton pump of plants was essentially similar to mitochondrial APAse. He also adapted electrophysiological techniques first developed for exploration of neuronals' channel properties to determine that pumps at vacuolar membranes exhibit kinetic responses to ion gradients that would not be predicted through biochemical means. Parallel to this, he discovered that vacuolar membranes exhibit electrically driven ion release. Using both electrophysiological and biochemical approaches, Sanders was able to establish for the first time in plants that metabolites can act as triggers for release of calcium, a cellular signal from vacuoles. Sanders established principles for befortification of cereal crops with essential human mineral nutrients and molecularly characterized calcium-permeable channels. Sanders also discovered and characterized the first and only yeast calcium channel and demonstrated how cell marking can be used to distinguish cell types for patch clamp studies. Sanders also had influence in the investigation into the roles of plant cyclic nucleotide-gated channels that were explored at an early stage of discovery and resulted in a major collaborative publication with another lab demonstrating a key role in plant bacterial symbiosis signaling. On top of his extensive discoveries, he has also written influential reviews on calcium signaling in plants, which have 3,300 combined citations on Google Scholar. To further his work on calcium channels, he then discovered that the channel is the major pathway for ion exchange across plant vacuolar membranes. Their speculations that the channel is involved in calcium-induced calcium release were proven for the first time in plants in work from Sanders' lab. He then established the principal molecular and cellular mechanisms for plant tolerance to manganese toxicity. Sanders has discovered the major mechanism of zinc accumulation in plant vacuoles, and more recently characterized the molecular properties of the transporter and showed how the transporter could be used for nutritional benefit for human consumption of cereal grains. On top of further collaborating with a Chinese lab to establish more generally the important role of zinc nutrition in rice, Sanders' current research focuses on how plant cells respond to changes in their environment and how they store the nutrients they acquire. In particular, his group work on how transport of chemical elements across cell membranes in plants is integrated with cellular signaling and nutritional status. As we progress through this video, let's now turn our gaze towards career. Sanders' research career began at the Yale University School of Medicine, first as a postdoctoral research fellow and then as a postdoctoral research associate. After a stint as a visiting research fellow in the University of Biological Sciences at the University of Sydney 1983, Sanders moved into the biology department at the University of York in 1983, first as a lecturer, a reader, a professor, also acting as the head of department. In 2010, Sanders moved to the John Innes Centre, Norwich, as director and group leader, establishing new collaborations with the Chinese Academy of Sciences. Now, let's dig deeper into awards and honors and unveil the hidden treasures it holds within. Sanders was elected a Fellow of the Royal Society in 2001. Throughout his career, Sanders has received a number of additional awards and honors, including fellowships, inaugural Fellow of the Royal Society of Biology 2009, elected to Royal Society Council China International Science and Technology Corporation Award 2021, Chinese Academy of Science CAS International Science and Technology Corporation Award 2021, Royal Society Liverhulme Trust Senior Research Fellowship, Nuffield Foundation Science Research Fellowship, James Hudson Brown Fellowship, Yale University Prizes, Corba Foundation European Science Prize 2001, President's Medal, Society for Experimental Biology 1987, Honorary Chairs, University of York present, University of East Anglia present, Agricultural Genomics Institute Shenzhen present. Thanks for being a part of this amazing journey. I can't wait to bring you more exciting content.